Now, remember what I told you. In a sense, quantitative genetics started without knowing Mendelian rules. Then it was formalized and given a foundation, scientific foundation, after Mendelian rules were discovered. But it was not until molecular markers came into the picture that you could actually start dealing with things that the theory was telling us. So I want to mention the important contributions of the school from the University of Edinburgh. In particular, this is Douglas Faulkner. He's the author of a very classical book in quantitative genetics, Introduction to Quantitative Genetics. I met him personally in Edinburgh in 1982 and then in 1998. I don't know if, he was alive still in 1998, but I met him in 1982. Very important book. And then two persons, Alan Robertson and Bill Hill. Professor Robertson passed away in 1987. He was very interested in using ideas of no less than Moto Kimura to arrive at the probability of fixation of an allele in a small population that was undergoing selection. So Robertson has a number of important papers on this matter. And this is Professor William Hill. He's probably getting close to his 80s. He's fortunately still with us. He's still alive, but he has retired. And maybe he's a bit less active now than he was a couple of years ago. But he made many, many contributions, including a range of papers on linked disequilibrium. That is something that became quite important in the search for quantitative trait loci. William Hill is a person that I admired very deeply. He was always very influential in my thinking. I always respected a great deal what he said, and perhaps it's come because I had to present a paper as a student in 1972, and I was able to derive, I was able to present all the formula, but I was never able to derive many of them, and I still can't. Then markers came into the picture as a consequence of the mapping projects. And you know what we have done, a lot of developments, and we have most species have full sequences, and the cost of sequences coming down and down and down and down. So the basic idea that came is that of relating variation at some loci called singular nucleotide polymorphisms, and try to relate variation at the level of markers with variation at the levels of the phenotype. So, for instance, there are markers encoding regions and regulatory sequences elsewhere, and the idea was to relate markers to phenotypic variation to see if they could be used either for agricultural breeding or perhaps for clinical practice in personalized medicine. So the first type of paradigm, which is very active in human genetics, is called genome-wide association studies, where they search for genomic regions that are associated with traits. The first study was done in Japan with macular degeneration. There are many recognized genome-wide association studies, and if you follow some of the papers like Plot Genetics, Nature Genetics, Nature Review Genetics, there is a great deal of emphasis. And there are some, they measure everything, especially diseases, but sometimes there are some traits that you may think are humorous, like how hard you shake the hands of others. Maybe in Japan is the angle with which you bow, the propensity to cry, and many other things. There is a real question as to what extent genome-wide association studies are helpful for practical purposes, both in agriculture and in medicine, but it's still a very active field. So this is a standard way of representing genome-wide association studies. Each of these points is a chromosome by chromosome, marker by marker scan of an association. Normally regression methods, linear or logistics are used. And because you do many, many scans, hundreds of thousands, you have to adjust for multiple comparisons. There's a problem called multiple testing. So there are very stringent significant thresholds, and when this threshold is exceeded, then you declare a significant association. Whether that association has a predictive value or not is a very doubtful issue, because statistical significance and prediction are not necessarily related, okay? Sometimes statistical significance lead to better predictions, sometimes it doesn't, and sometimes predictions are better using things that are not statistically significant. This is something that we know quite well in animal and plant breeding, but perhaps was not that well known to other scientists. And the funny thing about this is that this paper published in the Proceedings of the National Academy of Sciences, the senior author came from a Department of Political Science. And frankly, the political scientists are not very good at predicting things. Just think about the elections in the United States in 2016. Nobody was able to predict that Donald Trump would become president of the United States. So there is a second paradigm that has become very dominant in animal and plant breeding, that is called whole genome regressions. The paper was presented, published in 2001, and the idea is, instead of looking at marker by marker thing in the model, like trying to find QTLs, just use all the available variants that you have and put them all together in a model, and then use methods to temper this problem that are called P larger than N. There are methods to avoid overfitting and to convert these methods into good prediction machines. So genomic selection was first applied in dairy cattle in the United States and in the Netherlands in 2007, and now it's applied in dairy animals, beef animals, sheep, pigs, aquaculture, maize, barley, wheat, coffee, sugarcane, palm oil, rubber trees, forestry, etc. So it has become a very important and useful technique. So these papers suggested 3 methods, but then many people have extended the class of methods, which include many, many different methods of linear regression, and more recently methods of machine learning, and we will say a little bit about it later on. So to give you an idea how genomic selection works, you have to have some chip. In dairy cattle, now we are using chips of about 800,000 markers, and the number of animals that were genotyped in this paper was published in 2017, so the data are only up to 2016. At that point, we had a bit close to 200,000 bulls genotyped in the US, and about 1.2 million cows in the Holstein breed only. So now I wouldn't be surprised if about half of the cows in the US are genotyped, and we have about 9 million cows, maybe a bit more. In another paper, they estimated using genomics what have been the trends in these populations. So I want to bring to your attention the blue lines. This is the estimated genomic trend for milk production, this is for fat production, and this is for protein production. And genomic selection began to be applied in about 2007, so you see that a couple of years after the introduction of genomic selection, then the population started changing much more rapidly than before. And this is due to 2 reasons. One is because the predictions based on markers are a bit more accurate than pedigree-based prediction, but also because you can get estimates of the breeding value of animals much earlier in life, so you can make selection decisions and choose your replacement bulls, for example, much earlier than before. Under standard progeny testing, the generation interval was about 6 years, and now the generation interval is about a little bit less than 2 years in cattle. So we are turning generations much more rapidly. And in the right, we have what are called functional traits. This panel is the somatic cell count in milk. The smaller the number of cells, the healthier the cows are. So we are going in the right direction. This is the productive life, namely the number of days that a cow stays in the herd. So the cows now have a larger productive life, genetically speaking, and this is reproduction. In the Holstein breed, which is actually a machine of producing water, reproductive performance was deteriorating very severely, and the reason was that increased selection for milk production was making the animals less functional. So then when selection objectives changed to give more emphasis to reproduction and genomics came into the picture, then things started moving in the right direction. So it looks like we are doing things right, at least in dairy cattle, getting more productive animals that are healthier, that they reproduce better, and that they stay for a longer period of time in the life. One problem, but we still do not know how to cope with, is that of gene-by-gene interactions, actually gene by gene by gene by gene. The medical geneticists talk about gene-by-gene interaction, and I think it's a bit misleading because, as I said before, from a biological perspective, high-order interaction might be quite important. However, when you use statistical methods, you do not find much evidence of interaction. So the question is it because the methods are wrong, or is it because interaction is not important? Well, the answer, if there are, of course, many groups in the world that try to make sense about interaction by using these so-called systems approaches or structure or systems biology. This is one representation. So some people are repeating something that used to be said in the mid-60s in anthropology, and in Germany there was the theory of general system theory of Ludwig von Wernher-Lampe, that you have to take a look at the system as a whole and not at the parts. So some people are trying to use this systems approach, but once you come up with a diagram like this, it's very, very difficult to make sense out of it, even though there are just a few nodes in this system that in some sense are representing something related to obesity or hypertension or something like that. Now, this problem of why interaction doesn't show up statistically was investigated in a paper by William Hill, and they actually found that even for epistatic systems, most of the variance turns to be additive. So in a sense, these important interactions from a biological point of view, they are hiding. They are generating additive variance, so we will never be able to understand complexity using linear models, because linear models or standard parametric method most of the times will tell that interaction is not important or that it contributes very little to the variance. Yet we know that mechanistically it's expected to be quite important. So what's happening statistically? Well, what happens, if you remember your basic mathematics, in calculus, there is something called the Taylor series expansion of a function. So you have a function, you expand it by using a linear term, a quadratic term, a cubic term, an nth order term, and quite often, near this point of the expansion, the linear function does a very good job, and that is essentially what's happening. The linear model, this additive model, is giving you a representation that it locally is quite good. It's helpful to do predictions for a reasonable, in a neighborhood of time that is reasonably small, but then when things change, you have to adapt your regression line. But we still do not know all the surfaces, concavities, and convexities of the genotype to phenotype map because there are just far too many dimensions. So sorry about that. The fonts do not embed very well. So in essence, if you believe the statisticians are what comes out of most quantitative genetic analysis, then life is a straight line. And it's an operational thing. You go out to the street here and you look in this direction, that direction, that direction, and the earth looks flat, but it's not flat. So what I'm trying to convey here is the statistical methods are actually good to describe, good to predict, but not always good to explain. Just think about Galton. We still do not know why this regression bends at some point, which is about 67 inches in humans. So if we want to understand something like this, you have to cross that, okay? To understand something like this, there are better approaches, molecular genetics, for example. But of course, you have to reduce the system. You'll be very strongly reductionist. But given resources, given money, given time, sometimes you are able to decipher or understand important genes, such as a TP53 gene, which is one of the first battles of defense against cancer in humans. So people talk about genetic architecture statistically. Well, these are some beautiful piece of architecture. This is an Iraqi architect that did most of her career in London. This is an Uruguayan architect that constructed this bridge that is very beautiful when you look at it from an airplane. But if you go to Tokyo and you go to the Tokyo International Forum, which is a magnificent building, especially Hall G, it was done by this person that happens to be born in the same country where I was born, which is Uruguay. This is the famous Bilbao Museum by Frank Gehry, and this is the scientific city of Santiago Calatrava in Valencia, Spain. So this is architecture, and not how many bricks, how many steel, and how much hours of human labor it took. Architecture is the integration of all these things, and the same applies to genetics. Okay, so the bricks and the hours of labor and the kilos of steel or tons of steel are the linear regression method, but the architecture is something much more subtle. So for that reason, I do not think that we'll ever be able to understand the genetic architecture of complex traits, but we have to regard the method that we have in a modest manner. They can predict, they can describe, but they are not very good to explain.